Everybody, today we are going to discuss a important topic, that is Streptococcus pneumoniae, commonly called as pneumococcus. It has been already discussed in your class. It's a revision lecture for you all. As you all know, pneumococcus is gram positive lanceolate diplococcus, commonly called as Diplococcus pneumoniae. It differs from other streptococci, especially in its morphology, biosolubility, optocin sensitivity, and its capsule. They were first noticed in 1881 by Pasteur and Sternberg independently, producing a fatal septicemia in rabbits. Morphologically, they are typically small, slightly elongated cocci with one end broad or rounded, having a flame-like appearance, also called as lanceolate appearance. They occur in pairs, hence they are called as diplococci, with broad ends in opposition. They are capsulated and each capsule encloses one pair. They are readily stained by aniline dyes. They are gram positive in nature, and the capsule may be demonstrated as a clear halo in India ink preparations. Now coming to the cultural characteristics, they grow only on enriched medias. They are aerobic in nature and can be facultative anaerobes. The optimum temperature is 37 degrees centigrade and pH ranges from 6.5 to 8.3. On blood agar, after incubation for 18 hours, the colonies are small, dome shaped and glistening, with an area of greenish discoloration around them. This area of greenish discoloration is called as alpha hemolysis. This is generally seen in Streptococcus viridans. On further incubation, the colonies are flat with raised edges, and also there is a central umbonation. Umbonation means umbrella like in shape, so that the concentric rings appear like draught man colonies or carom coin appearance. Under anaerobic conditions, the colonies on blood agar are surrounded by a zone of beta hemolysis due to oxygen labile hemolysin O. On liquid media like glucose broth, the growth occurs as a uniform turbidity. Biochemical reactions include the pneumococci generally ferment several sugars forming acid only. The fermentation is tested in his serum water or serum agar slopes. The fermentation of inulin by pneumococci is a useful test that differentiates it from streptococci. They are bile soluble and are catalase and oxidase negative. They are delicate organisms and are readily destroyed by heat and antiseptics. The toxins that are produced are oxygen labile hemolysin, commonly called as pnemolysin, and leukocidin. Both of them are weak toxins and their role in infection is inconclusive. Pathogenically, between 5 to 70% of normal human adults harbor Streptococcus pneumoniae in their throats because it is a commensal and many of the commensal pneumococci are non-capsulated and avirulent. It also causes bronchopneumonia, bronchitis, sinusitis, otitis media, meningitis, endocarditis, suppurative arthritis, peritonitis, and bacterial meningitis. Lab diagnosis is done by various specimens like sputum, blood, CSF, the acute phase of lobar pneumonia in which there is rusty sputum and it contains a large number of pneumococci. They are demonstrated by Gram's staining, also it is inoculated on blood agar plates at 37 degrees centigrade in the presence of 5 to 10% carbon dioxide. The growth occurs after overnight incubation if the sputum is not available. Then serum coated laryngeal swabs, as in case of infants, may be used for culture. If the suspicion of pneumococci is scanty or less, isolation may be done by intraperitoneal inoculation in mice, and the inoculated mice dies in 1 to 3 days, and the bacteria can be demonstrated in peritoneal exudate and heart blood. In acute otitis media, the pneumococci may be seen in fluid aspirated from middle ear. In case of meningitis, Gram's staining of CSF may be done. The diagnosis is confirmed by further cultures. So this is all for today. This I have just revised your lecture in very brief. If you have got any query, you can all ask me on my WhatsApp number. I think everyone is clear. So thank you.